Unfortunately, we lost those previous slides, but hope you all remember. So the next example of in sort of an integrated system. This is another example where the portability of the microfluidic device makes it useful. This is a cell counter for HIV diagnostics. So there are 33 million people in the world which are infected with HIV, and a lot of it is in the developing world. And unfortunately, the developing world is also where they have fewer resources to detect it. When you're trying to prevent the spread of a virus or spread of any type of pathogen, the very important thing is early diagnostics. Right? So you can treat, you can make sure that whoever is infected doesn't spread it to other folks, and so on. So a lot of folks in the microfluidics field were interested in how can we do HIV diagnostics. This is an example of an integrated system. This was published last year in Science Translational Medicine. It uses some of the technologies that have been developed earlier by other researchers. It gives you an idea of what can be done with this device, microfluidic technologies. So this integrated system counts CD4 cells using electrical and optical techniques. And it also takes advantage of microfluidic techniques to separate the cells. So let's take a closer look at this system here. So the procedure begins with a whole blood sample, as we can see here on the right. So the red blood cells are first lysed. So what we're looking at here is this left diagram here. All right, the blood cells... Doesn't seem to like, there we go. The red blood cells are lysed. Then there's a quenching step. Then they do electrical cell counting at the entrance and exit of a chamber. And then within the chamber, the cells are captured on antibody-coated posts where they are imaged through a microscope. So let's look at some of the details of that right now. So again, starting off with a whole blood sample. Okay, you can see here on the top left. Now, a typical blood sample has many different cell types in it. Right? There's leukocytes, erythrocytes. You can see here. There are CD4 cells. This is a type of immune cell. And the count of those cells indicates whether you are likely to have HIV or not. So this is CD4 counting. So essentially what we're doing here from the physics perspective is we are just counting a certain cell type. So you put in the cells here on the upper left. And then right below that you put in a lysing buffer. Lysing means that it actually tears up the cell membrane so that the cell basically breaks up. All right, what they do here is that the lysing buffer actually selectively lyses the red blood cells, the erythrocytes, and also, well, it selectively lyses the red blood cells more than it lyses the CD4 cells and other leukocytes. So they're taking advantage of that property. They add the lysing buffer here, and then they run these components through a microfluidic channel. Now, as these things are flowing through the channel, they exist in that microfluidic channel for a very specific amount of time. Now here's something that happens. If you keep the lysing buffer in there for too long, all the cells will lyse. Okay? So you want to keep the lysing buffer in there for a very specific amount of time. The nice thing that you can do with microfluidics is that if you have the cells coming in at a specific flow rate, the length of the channel will determine how long the interaction happens. So what's happening here is that you add the lysing buffer at this portion of the channel, but here you're adding a quenching buffer. Quenching buffer means it stops the reaction. Okay, so the lysing happens only for the time that the cells flow through this section of the channel, and you can control that down to milliseconds. Very precise. You can start the reaction and stop the reaction right away by adding the quenching buffer here. Now you can see that most of the red blood cells, pretty much all of them, are lysed. There are some of these cell types which are also lysed, but a small percentage of them. these continue to flow down the channel. Okay, the cells come in here, and you notice these 3 black lines there. Those are electrodes. Okay, these electrodes are actually doing cell counting. All right, there's this a principle called impedance spectroscopy. I can just back out here for a second. So, one note. Oh, we saved all the images, but unfortunately no recording. Okay. So, the channel looks like this, and you have electrodes. One electrode here, one electrode here, and one electrode here. Let me redraw the channel. These electrodes are actually exposed to the inside of the channel. So as a cell goes by, let's say a cell is continuing this way, eventually it gets right here. Okay, and this is connected to an electrochemical circuit which measures the impedance between these electrodes. Okay, you can do this impedance measurement with just 2 electrodes. With 3 electrodes, you have you can account for you can have like an auxiliary electrode, working electrode, and a measurement electrode. So it makes things a little bit more accurate. But the point is, you're measuring the resistance of just this portion right here. When a cell goes by, it changes the electrical resistance. So this is where you're using electric fields to actually count the number of cells as they go by this section of the channel. Again, the advantage of doing this in a microfluidic device is that the cell is the same size as the channel. So it really is covering up most of the channel. So it causes a large resistance change. So going back to this area, then you have the impedance cell counter coming at the entrance. Right, this is the entrance counter. You also have one at the exit. In between them is a capture chamber. Now this capture chamber is a small microfluidic chamber that has posts in it. The posts are those like little blue circles that you see over there. The posts are coated with antibodies, and those antibodies capture the cells of interest. They capture CD4 cells. Again, for those of you who aren't chemists, biologists, which is most of you, I would say, antibodies can be coated on posts. Antibodies are molecules, biomolecules. They're on the surface of CD4 cells. There are these things called receptors, and those receptors match up with the antibodies, and that causes the cell to stick to the post. Okay. Now, where's miniaturization? Where are you taking advantage of miniaturization here? Well, when you go to, if you shrink down the size of the device, the walls, you're always very close to the walls, right? If your device is becoming, we're in this room, right? If we shrink down the size of the room, I'm going to be closer to the walls. If I'm closer to the wall, there's more likely that I'm going to hit the wall if I'm being thrown around this room. You get the idea? So the surface area to volume ratio increases in microfluidic devices, in microscale. And so that increases the probability that the cells are going to collide with the posts. If the cells collide with the posts and the posts are functionalized, then the cells will stick to them. The cool thing about antibodies is that they're specific. Now this part doesn't have anything to do with miniaturization, it's just really cool chemistry. Antibodies are very specific to a certain cell type. So they will capture the CD4 cells, but they will not capture like the other leukocytes. So when you're flowing from right to left, what will happen is that the leukocytes will flow through, the CD4 cells will be captured. There might be a few leukocytes that end up getting in there, but you get the idea. So you can, when you image this, you can actually do a count of the CD4 cells that way, relative concentration, and you're actually capturing them for further analysis later on. Now you have an exit counter here on the left, as I mentioned. So what's going into the chamber is a combination of CD4 cells and leukocytes, so you're going to get a certain count, and what comes out of the chamber is only the leukocytes, so if you subtract the two, you'll get a count of the CD4 cells. Right, so this is an interesting way to get a CD4 count in a way, and you're working literally from whole blood. So this is an integration of multiple functions on the chip. So this type of chip incorporates some electrical components. It incorporates some chemistry on chip. It incorporates some biology, the antibodies. You also have some imaging, right? And it also incorporates, to do the detection, the measurement circuit, you have to have some circuits in there too. So there's really a lot of things that go into building a BioMEMS device. And this is why, you may wonder why it's such an interesting field, is because from our perspective as engineers, it's fun because you actually get to learn about, you get to learn a little bit of biology, you learn a little bit of chemistry, you work with a little bit of electrochemistry. So you really are engineering interdisciplinary systems. It's fun because you get to learn about all these different fields. And while it might seem intimidating at the beginning, the more you learn about it, the more you see that there are, the basic concepts are actually quite, quite understandable, even if it's outside your field. The challenge is that the devil is always in the details. You will never know how difficult what might seem to be a very simple biological assay is until you actually try it in the lab, and then you realize that why it takes a biology PhD student like 8 years to graduate. Okay, so this device does the accurate CD4 counts in about 20 minutes with a single drop of blood. So notice that microfluidic device is using very small amount of blood. So that's also nice for the patient because you can get by with just small pricks instead of the big needle that they use when you're donating blood or when you're having like 100 different tests done at your doctor's office. Let's take a short break since we are gone for a while. We'll come back to this, and hopefully we can finish up this module at the end by the end of class today. So we'll just take a 5-minute break. We'll pause this. All right. So I'm going to talk a little bit about the commercialization of lab-on-a-chip technologies, just to give you a flavor for what is happening on the industry side of things, and also to show you that these technologies really are, they're no longer just whims, laboratory whims. They actually are being commercialized and used in a lot of very interesting applications. I'm not going to go over this entire slide, but just want to touch on a few different ones. One of the earliest companies in microfluidics was Caliper Life Sciences. They made lab-on-a-chip systems for diagnostics and molecular testing. A lot of electrophoresis systems. They were one of the earliest companies. They were later acquired by Perkin-Elmer. So they started in 1995. As I told you, the microfluidics field also started off in the early 90s, early to mid-90s. Another well-known one is Stephen Quake's company, Fluidigm. They are commercializing those chips that we saw, earlier, the very high throughput devices that had thousands of valves on chip, thousands of individual chambers. They're using them for high throughput biology. They have used them for single cell biology that we talked about earlier, but unfortunately was not recorded. So Fluidigm has a lot of commercial systems right now for genotyping and sequencing and other types of assays. They were founded in 1999. Cepheid was founded in 1996, also focused on portable diagnostics. Some of these companies, Micronet Microfluidics, Dolomite Microfluidics, they are focused on manufacturing custom microfluidic devices. Now, a lot of microfluidics researchers, a lot of companies, they don't have foundries. Foundries is where you actually do the microfabrication. So these companies provide those types of manufacturing services. And they also have some individual products of their own. They actually have catalogs of microfluidic chips that you can buy. Over time, like when the microfluidics field first started, every microfluidic chip design was custom. Okay, now over the last 10 or 15 years, you realize that actually many of the chip designs are, something that I do in my lab today is probably the chip design will be very similar to what someone else did, 5 years ago for a different purpose, right? So there are a lot of standard chip designs out there that are being sold by companies like Micronet, Microfluidics, Dolomite Microfluidics, and so on. So a lot of microfluidics work right now has become more accessible to a larger population simply because you can go out and just buy these things now. A lot of research still requires custom devices. So we talked a little bit about the field of droplet microfluidics. There are some companies that are doing quite well in that area. Raindance Technologies was founded in 2004. They're working on droplet microfluidic devices for genotyping and sequencing. There is a company called, what was the name of it? Can't remember the name of the initial company, but it was acquired by Bio-Rad. They were doing digital PCR. I haven't talked about digital PCR, but it's a new form of PCR which allows you to accurately count the number of DNA molecules in a given sample. They started off in the early 2000s, and they were bought out by Bio-Rad just a few years later for $150 million or something like that. The companies who really make it in the biotech field, some of these microfluidics companies, they really hit it big. And there have been countless other companies which have started and have failed. It's an early, like in terms of commercialization, it's still a relatively young industry. And over your careers, you will see many more companies flourish and many more companies fail in this area. You'll probably see consolidation of companies, like merging of small microfluidics companies. You'll see large biotech companies like Roche, Illumina, buying out smaller microfluidics companies for their specific technologies. And hopefully someday you all will develop microscale devices on your own and then sell them to large companies and move to Thailand and stay there. Another example of a buyout recently was Illumina. Has anyone heard of Illumina? Has anyone heard about Illumina, the company? Okay, they do genetic sequencing. They make the machines to do DNA sequencing. DNA sequencing is, remember I was talking about DNA is your genetic code? They can actually read the individual parts of your genetic code. You're literally reading the blueprint. We'll talk about sequencing later on. But this is a very powerful technique in the life sciences. Illumina bought out a microfluidics company called Advanced Liquid Logic up here. So Advanced Liquid Logic was making electrowetting-based systems where you can use electric fields to control droplets and move them around, merge them together, split them. We'll talk about some of their technologies later on in the course. It turns out that technology was very useful for preparing the libraries used in next-generation sequencing. So Advanced Liquid Logic was acquired by Illumina just last year or the year before. I believe it was for an undisclosed sum, but it is quite exciting for the microfluidics field to do that. They were, oh, here we go. They were acquired in 2013. Yeah, so that was maybe a year and a half ago. So a lot of movement in the fields. If you're interested in learning about commercial devices, you can read this article that was published this year. Well, now it's 2014, so it was last year. It's Commercialization of Microfluidic Devices. We can go over a few examples of commercial microfluidic systems. Agilent Technologies was one of the first players in the field. I mean, they acquired a company. I believe it was. I can't quite remember if they acquired a company or they developed the technology on their own. But the types of devices that they sell We talked earlier today about electrophoresis, capillary electrophoresis, which allows you to separate DNA molecules by size. Capillary electrophoresis can also be used to separate other analytes by size, like metal ions in environmental samples. You can separate those by the size of the metal ion. So you can determine the concentrations of different metal ions in a sample. You can analyze proteins this way. So Agilent started selling chips for capillary electrophoresis, where you could put in the sample, you could do the electrophoretic assay on chip, and they have optical readouts where you can get these electropherograms to analyze a sample. Some of these chips have gels inside of them. The gels actually help improve the separation resolution, and some of them actually operate without any gels at all. So these types of chips are used for diagnostics. The picture here shows just the microfluidic chip. These microfluidic chips are typically consumables because they're so small they can be thrown out afterwards. You have these consumable chips that are put into like a larger desktop-like device, which reads the chips, it performs the assay, and so on. Raindance Technologies has, I mentioned before that they do droplet microfluidics. They do something called digital PCR, which I won't go into the details of it right now. But it's one of those types of biological assays. It allows you to count the number of DNA molecules in solution. It uses droplets because the DNA have to be encapsulated into really small chambers where on average, statistically, there's like one DNA molecule per droplet. Okay, one or fewer. By doing this, they can actually count the number of DNA molecules. They do PCR on each one of these droplets, and they look at which droplets fluoresce. The number of droplets that fluoresce tells you the number of DNA molecules in the solution. Very accurate, very precise way of doing DNA counting. They're becoming very, very popular right now. Raindance Technologies and Biorad was their competitor. They also do digital PCR. Very exciting and growing area right now. Fluidigm Life Sciences was, as I mentioned, Stephen Quake's company. They sold these PDMS devices that had a lot of chips and a lot of valves and pumps on chip, a lot of individual chambers for doing single-cell analysis here on the left. And then they also do digital PCR. Although their digital PCR products have been superseded in some ways by Raindance's technologies and also Biorad's. These guys are using droplet-based digital PCR, which allows you to get 20,000 to 100,000 different chambers. With the digital array, it allows you to get maybe hundreds to maybe a few thousand chambers. With digital PCR, there's a lot of advantage to doing things. The more chambers you have, the more precise the measurement becomes. So, but Fluidigm still has the advantage and the techniques for single-cell analysis. This company is doing very well. Perkin-Elmer sells the another on-chip electrophoretic separation system, and there's a link to a video here. So why don't we just take a look at that? Hopefully the link is still working. Oh, let's try copying the link. Try it once again. Maybe it'll show up. Okay, we'll come back to that later if we have time. I want to make sure I get through the rest of this material here. A new area. This is a very recent area, next generation DNA sequencing. Well, some of the next generation sequencers have been around for a little while, but there are some newer technologies which have come out just in the recent years, which is making things very exciting. So we talked a little bit about DNA sequencing. I basically said that DNA sequencing allows you to read someone's genetic code. I'm not going to go into the details of that right now, but DNA sequencing is a pretty complex chemical assay. Have any of you have heard of the Human Genome Project? Some of you? Okay. This made a lot of news in the early 2000s, the mid 2000s, and it was the first effort to completely sequence a genome. So our DNA, we have our genetic blueprint. The blueprint that's used to create us is called the genome. Okay, we have DNA inside each one of our cells. Each cell contains the entire genome. It has all the DNA. The genome consists of 3 billion base pairs. Okay, there's it's sort of a language. You can think of DNA as having a language. There's 4 different types of bases: A, G, C, and T. They're 4 different molecules, and the DNA is a strand. I'm going to make just a very crude diagram here so you get the idea. The DNA is composed of a backbone like this, and along this backbone there are different bases attached to this backbone. There are 4 possible bases: A, G, C, and T. All right, and DNA is just this molecule. This is called a phosphate backbone. And these are called bases or nucleotides. The sequence of these bases can be read by a DNA sequencer. Okay, you imagine that there are 3 billion bases in your DNA. Reading each one of those bases would take an inordinate amount of time. It's also quite expensive. 3 billion. If you did that at a rate of 1 per second, you can kind of figure out that it would take many, many years to do that. That's not the case right now. You can read them actually much faster nowadays. I wish I had a slide on this, but if we were to look up in Google, maybe we can. Let's just come back to this. The initial Human Genome Project, it cost $1 billion, $1 billion to sequence one genome. Very, very expensive. Okay, so obviously that's not practical. It's of scientific importance, but not practical. Now they've gotten to the point where Illumina, this company that I mentioned to you, they have just in the last year or so, they claim that they can do genome sequencing for less than $1,000. They went from $1 billion to less than $1,000. This $1,000 mark was very important from a commercial and medical standpoint, because now what that means is less than $1,000. That means we can actually sequence everybody's genome, and that healthcare companies will actually consider paying for it. Now, if everybody's genome is sequenced, you can imagine how that would transform medicine. Okay, I would have my genome sequenced close to when I was born, and they would say that, okay, you have this defects in your DNA, so you are at a high risk for these diseases. Right? It could be used for diagnostics. It can be used for, if you have a high risk for cancer, for example, there's a gene called the BRCA1 gene that's related to breast cancer. If you have it, you have a 75% chance of getting breast cancer. Okay. So if you have certain genes, then what you can do is you can make sure that you're getting checkups regularly. You get mammograms regularly in that case. You can make sure that you're having a very preventative lifestyle to avoid those diseases. You're going to be more vigilant, right? So this whole era of personalized medicine will become possible due to DNA sequencing. Okay. So next generation DNA sequencing, the first generation of sequencing technologies used something called Sanger sequencing. That was very expensive. not very scalable. There are next generation DNA sequencers, the second generation, which is shown up here. Right, this is the Illumina optical sequencer. It uses a microfluidic chip actually, where the again, I'm not going to go into the details of how it works, but the DNA is actually loaded into each one of these wells here. The samples flow through here. Illumina uses an optical technique where you do this assay called sequencing by synthesis. If I go into the details of it right now, it might become quite confusing. But it basically uses an optical technique. There's a flash of light that occurs when a certain base is incorporated into the genome. They try to match nucleotides with complementary bases in the genome. If this doesn't make sense, don't worry about it. But this technique allows them to sequence DNA samples at a manageable cost. I'll say it's still quite expensive, but not nearly as expensive. That's the $1 billion for the Human Genome Project. Life Technologies and Ion Torrent was a company that was ultimately bought by Life Technologies. They use semiconductor sequencing. They have 6 million individual wells. Okay, we talked about wells where each well is chemically isolated. They have 6 million different wells where they put DNA strands into each one of those wells. And then they sequence each and every one of those strands. Okay, and the way they sense what type of bases, whether it's A, B, C, A, G, C, or T, is by using a sequencing technique, or a sequencing by synthesis technique, and which also requires a semiconductor sensor. So they have a whole array of wells, and they have a semiconductor pH sensor in each one of those wells. So 6 million pH sensors they have in this chip that's about a few centimeters by a few centimeters. This is really an amazing, quite an amazing technology. It's actually the marriage of microfluidic technologies with semiconductor technologies with chemical sequencing. The guy who who came up with this technique is a genius. His name is John Rothberg. He actually figured out this optical sequencing, and then later he went on to invent this technology also. It's quite an amazing story if you read about Jonathan Rothberg. Great stuff. Ion Torrent predicts that their semiconductor sequencing technology will ultimately become cheaper than Illumina's, but that still remains to be seen. A very exciting but yet very unproven technology is the Oxford nanopores technology. Now, when I showed you this diagram earlier, like where you have this DNA backbone and then you have these different molecules attached to it. The most direct way of reading that DNA sequence would be to just, if you had some ways of reading each one of those molecules directly. So Oxford nanopores technology is quite interesting if it works. What they do is they create a really small pore. They use microfabrication techniques, and they create a nanopore. I'll just tell you conceptually what's happening. And this DNA molecule translocates through that nanopore. As it translocates through that nanopore, these different bases will have different sizes. Okay, so I'm going to exaggerate this here, but imagine that if you have a small, one of the bases is small like this, and another one is slightly larger like this. The electrical resistance across that pore is going to change because the size of the molecule that's blocking it. So that sort of principle is shown here, that if you have a nanopore suspended in a membrane as the DNA molecule is going through, just a single DNA molecule, these nanopores are like a few nanometers in diameter. As the different molecules DNA bases are translocating through the nanopore, the electrical resistance of that nanopore will change accordingly. And supposedly, you could then just read out the sequence of the DNA directly. If this were possible, this would allow you to make a sequencer that is this tiny and can be plugged into the USB port of your computer. Okay, they have these devices out, the mini ion sequencer. However, the initial studies that some of the scientists in the field have said that it's not quite accurate. It's not as accurate enough as it needs to be. It's not as accurate as Illumina system. Illumina systems cost, millions of dollars. this type of system could make it affordable to, you could have one at your home, supposedly. So these are all examples of where microfluidic technologies and microscale technologies are really transforming medicine. And you will see these devices, this slide in particular, these next generation sequencing technologies will really change medicine and how healthcare works over the next, over the next decade. More examples. Looks like we may not get through all these today. Part of the reason I am showing you these different areas within the microfluidics field is to also give you an idea of what your projects, your semester projects can be. It's helpful if you kind of have an idea of what's happening in the field to kind of see which area is of interest to you, and you can explore those further. But that doesn't mean that you have to do one of these topical areas. In fact, I suggest that you just find a topic that's of interest to you, maybe close to the research that you're doing in the lab right now as PhD students, or just a topic that you're interested in. This particular area of microfluidics and BioMEMS is called organs on a chip. The motivation for it is shown up here. Okay. So when we do drug testing, I showed you guys the microplates, right? With microplates and petri dishes, you put the cells on there. You might put a drug or some type of chemical on there, and look how the cell responds. Is it going to kill the cell? Well, then we better not use that drug in our next, in a product that we're going to, in a shampoo, for example, that we're going to sell to consumers. That's called toxicity testing. In drug discovery, they'll look to see if this is affecting the cell, if it's causing the cell to secrete more of a certain, more insulin, for example, if you're looking at diabetes drugs. Just giving you some examples here. The problem is, when you do tests on cells in a petri dish, that's called an in vitro model. Cells in a petri dish do not behave the same as cells in the body. In the human body, you have a cell extracellular matrix. It's kind of a glue that sticks that holds the cells together. You also have the delivery of nutrients through blood vessels, right? Whereas in a petri dish, the situation is quite different. So you have diffusion of nutrients through the walls of capillaries and so on. You have multiple cell types that interact with one another and send messages to one another. So the in vitro model doesn't accurately predict how a drug or a chemical will behave inside body tissues. So this field of organs on a chip the idea of it is to create environments which closely mimic things that are happening in the body. So cells can be cultured in microfluidic chambers which emulate the shape and the function of tissue, providing an in vivo-like model for testing therapeutics. An example of this is Harvard's lung on a chip. If we look at the structure of a lung, some pictures of them are sort of shown here. All right, in a lung, you'll have a membrane separating the blood vessels on one end. Remember, in your lungs, if you remember from anatomy, physiology, the oxygen in the air diffuses into your blood. So your blood becomes oxygenated there. The structure of one of the alveoli or the sacs in your lungs looks something like this, where you have capillary blood vessels on one end. So this is a blood vessel wall. You have a membrane, or you have something separating them. And then on the other side, you have the lung cells, and this would be exposed to air. Right. So in addition to that, this membrane or this structure is constantly expanding and contracting as you breathe. Right? In order to accurately model that, if you want to test things like the respiratory effects of environmental toxins, if you want to test aerosolized therapeutics, like asthma inhalers and things like that, things that go directly into your lungs, you need to have a system that models what the lung looks like. So this field is taking off right now, or has been. There are systems for modeling lung on a chip, modeling guts, on a chip. There's a paper by a colleague of mine who was doing modeling a blood-brain barrier. There are drugs that circulate within your blood, but they don't go into your brain because there's actually a barrier that separates them. So they created a model of that in a microfluidic chip. This was just a couple years ago. So these types of chips actually have a lot of, obviously they have a lot of utility in drug discovery and toxicity testing. This is an example, let's see. I think this company's name was Emulate, if I'm not mistaken. I don't quite remember the name of the company, but the Harvard and the Wyss Institute, they actually commercialized this technology. It's a simple microfluidic device. It looks rather simple, but what it does, it emulates the structure of a lung, and they've used that for testing these types of things that I just mentioned, environmental toxins, airless therapeutics. So what they do in the microfluidic device, they create a membrane, and they flow in cells. They flow in one type of cells on the top half of the membrane, then they flow in another type of cell on the bottom half of the membrane, effectively recreating this type of environment. In addition, what they do to that is that they actually have two adjacent PDMS chambers on the left and the right here where they're applying a cyclic vacuum. The cyclic vacuum emulates the stretching and contraction of the lungs as breathing is happening. So this models the epithelium and the endothelium, blood vessel on this side, gas on the other side here. Okay, and this is being commercialized. For those of you who are interested in organs on a chip, there's a nice review paper by one of the leaders in the field, Sangeeta Bhatia, who's at MIT, Harvard, and then Donald Ingber, who's the founder of this place called the Wyss Institute. It's a pretty high-profile institute. So this actually was just published in 2014. It looks like we are out of time. We will continue again with this module. I'll go over some more examples of different areas of microfluidics which are of interest. We'll start with microfluidics for cancer diagnostics next time, and we'll definitely finish up this module next class and move on to scaling laws. So I'll see you all next Monday.